Dr. Rendeiro was an alumni of Cornell. Actually, was here for a while in Olivia Elemento's lab, and now has his own laboratory. It's done a lot of the preliminary, really pioneering work in spatial transcriptomics, spatialomics. I had the great pleasure to work with André during the pandemic a lot. We were all, traumatized by the pandemic, but wanted to develop as many methods as we could to better understand the disease, and did extraordinary work at the time, and has continued to do so now. Has his own laboratory, and you can see today at the Austrian Academy of Sciences at the Research Center for Molecular Medicine. So, Dr. Rendeiro, welcome. Thank you for taking time to speak to the class. I think it's evening there. Hopefully, not too late there. And thanks for joining us. And take it away.
Thank you so much. So happy to be here. It's not too late. I would like to keep this as interactive as possible. So feel free to interrupt me for discussion or for question. But otherwise, there's also time at the end. If you don't, I will also perhaps give you some rhetorical questions. So one of them we can start already now because depending on people's background and experience and interests, the answer to this question can be actually quite a lot. If you had a way, or if you had to choose a way to distinguish two cells or two cell types you have, in culture or in an organism, what would you choose to measure, right? This is very central to everything we do in biology and biomedicine. And different modalities give you different types of readouts. Of course, there'll be people nowadays that would say, we'll combine, have multiple ones, and that would be probably a very good answer. But I today want to argue, or rather demonstrate, if possible, that images are a very good way to capture the complex and heterogeneous systems that we're interested in studying. It has really single cell resolution, natively, let's say. It is quantitative and it can be multiplexed, so we can measure multiple aspects of the cells that we're interested in. And really, it shouldn't be that revolutionary to say this, but I think we forgot a little bit that microscopy and sort of our ability to look at tissues is really the original single cell method, as Anne Carpenter very eloquently said a few years ago, and then really everybody is starting or has since realized that. So today I would like to guide you guys through some of the technologies and questions we are trying to address with this type of data, starting with histopathology. So the good old way of looking at tissues and understanding what's going on with our physiology. So we'll have a case study, which is research done in my lab right now about aging from a histological perspective. And we'll also talk about multiplex methods of imaging, which I will talk about a study that Dr. Mason already mentioned on COVID-19, but also give a few sort of opinionated hints. Really, I won't spend a lot of time on that, on where I see frontiers in tissue biology. Which, spoiler alert, I already have them there. Some of them are really not surprising, but I think they're things that I think once put together will really revolutionize how we do and understand biology. So let's get started with some histopathology. And starting by appreciating that really the invention of the microscope opened up a whole new world of exploration on how we understand ourselves as a species, when we understand the world around us and our ability to see beyond what the human eye can. And it really was foundational to the start of disciplines that led the development of biology as we know it. So microanatomy, histology, physiology, embryology, and more. Really our ability to appreciate the world that we're looking at. But when we talk about tissues, we can't simply just place all the time, at least a specimen on a microscope and look at them, or we want to get a little bit more resolution, more detail on what we're looking at. So maybe you might know about this pipeline or this protocol to prepare tissue for histopathology, but I think it's maybe good to cover some bases that we need to have a way to keep the tissue in a way that does not deform when we try to section it. We need to section it to get light to pass through and to see the microscopic characteristics of the tissue. So a fixative is first applied. Usually the sample is dehydrated to get rid of lipids and to be more transparent. To stop also, the first step already starts a lot of chemical reactions that are happening. It will be cleared and it will be embedded usually, or at least for histopathology in a paraffin block, for example. And then it can be sectioned in a serial manner using a microtome. Nowadays, this can be done in a semi-automated way, and it causes sections that are 5-6 microns slim. That's kind of the standard. It can be 1 micron, for example, for very special applications. But this is kind of a very well-established way to prepare tissue for microscopy in general. If we have the specimen that then has no color, has no contrast for us to see, we need to apply some staining. So usually hematoxylin and eosin, so the H&E staining, which you'll hear a bit more in a second, is very standard, but there are more specialized stains for tissue. There's a lot of historical background for their development is actually quite interesting, so you can look more into it. But there's also an ability to capture some molecular granularity in these data, for example, using very basic immunohistochemistry, which basically we have a specificity to the certain epitope, apologies, that is conferred by an antibody, and then a colorimetric reaction, for example, or a reaction that is done in situ to reveal where the epitope is. This is all good, but the modern or a modern pathology lab nowadays incorporates several of these aspects in a more modern way. So, for example, the sectioning, as already mentioned, is semi-automated. The staining can be automated. The scanning of slides, that I'll talk about a bit more in a second, can also be automated. But a big component of it nowadays is also how we manage this data. How we manage the flow and how do we track the flow of samples through a laboratory. How do we manage the data once it's digitized, and how you manage things like storage and access, etc. So there is a lot to it, and fortunately we are quite happy to have this infrastructure a little bit more standardized nowadays in most places to work as we do. And when I say we, is maybe the field that I'm getting more into right now, computational pathology. So here's an example of kind of a state-of-the-art, even though things progress very quickly, a state-of-the-art model for histopathology, so-called the foundational model, because it's trained on a very large set of data in a self-supervised manner, which is also an important aspect. So in this particular case, it can also be a multimodal model, which means that it understands about the morphology of tissue, so a vision component of it, but also it has been trained on pair captions of these images, which, for example, come from a pathological report that goes to an oncologist, for example, from the pathologist that says what is in the image of clinical relevance or that is helpful for diagnosis. So the development of models like this has been very accelerated in the last few years, very few years, 2-3 years, and it's really opened up many more possibilities and avenues to explore this type of data in a completely different manner than what was usually, traditionally, perhaps in more following the field of computer vision. You would engineer some features that come from the image based on prior knowledge or biology that you would know, or maybe you would try to train on a data set that has very precise clinical labels. But a challenge was always the generalization of these models and how you can reuse them in practice. And this type of pre-training in a self-supervised manner, and also in particular leveraging the text modalities, really opened up a possibility of using a pre-trained model and fine-tuning only in very rare cases. So let's get a bit more into it. And to do that, I will introduce a data set that we've used now in the case study I'll present to you soon. So this is actually a data set that probably is known to a lot of people in the audience or in general to the genomics community because it was acquired to understand the relationship between genotypes and tissue-specific expressions. It's a GTEx cohort, and in which people were interested in understanding how variability at the genetic level conditioned the expression of healthy tissue in our body. So there were about 1,000 individuals for which there was a whole body donation, so up to 29 organs and 40 tissues were collected. And these were healthy individuals that were a representative population that was healthy, that did not die from terminal disease. And there was a rapid autopsy program in place to collect these tissues. And as already mentioned, the emphasis was for very, very long time on the genotypes and on the transcriptomes and a tissue-specific manner, trying to understand that relationship. But there were also images for these same tissues available for the longest time since 2012. Well, it turns out that back then, no one really knew what to do with these images, and they also did not have very clinically relevant labels, because as you can see in this example, we're looking at the whole slide image of the spleen. There is a little text here that says what's in the image. Maybe it says something like 2 pieces of tissue. One is 6 millimeters, the other is 2 millimeters or something like that. There's some vascular congestion. And this free text was also quite hard to work with. Nowadays, thanks to LLMs and also multimodal models, like I mentioned, we can leverage this much better. So this data set wasn't really used so much to study what I'm particularly interested, which is aging. So we know, thanks to decades of research, what's happening at the cellular molecular level to some degree in our tissues, but how does those changes lead to the progressive loss of physiological ability as we age is really not clear. So we thought this was the perfect data set to start addressing that from a very fundamental perspective. What is the composition of tissues? How does it change with age? What is the broader organization, architecture of the tissues as we age? And so at the time, we started this project in 2022, so soon after I left Cornell to come to Europe. And at the time, these foundational models weren't really out there yet. So we actually fine-tuned a lot of different vision models to extract rich quantitative features of morphology of tissue. Because most models that were available, that was another issue, that most were not publicly available. But the ones that were available were mostly for oncology, and we wanted to have features that represented healthy tissue. So later on, we realized that if you take a ImageNet pre-trained model, for example, something that has not even ever seen tissue, but just learned to classify a table from a chair, from a person, from a plane, we can also extract decent feature, but these are nicer. So what we saw is that if you collapse all the morphologies you see in a whole slide image of these 40 different tissues from these almost 1,000 individuals, so we are looking at 25,000 whole slide images. This is roughly 160 million tissue locations that we have passed through these models, that we recover. Unsurprisingly, that different organs have different morphologies and are easily distinguishable. We have organs like the brain that have different pieces of tissue. So also, for example, here is cerebellum versus the cortex, or different types of artery, the coronary, the tibial, and so on. For example, breast and adipose tissue are not as distinguishable as other organs, and so on. The GI tract are also more similar. This is interesting, and it's great that we recover this. Means that the data is of good quality, we can work with it. But the real realization we had that this data can be useful for our purpose of studying aging was when we also saw that within each tissue there was an axis of variation that most often was associated with age, or the strongest explainer of this variability between individuals within an organ or within a tissue was age, when we compared it to multiple factors. And by the way, we also, when we pre-trained these models, a lot of the work was on balancing the data set correctly and trying to make sure we have the, age brackets that are balanced, sex that are balanced, the amount of subclinical pathology in there, and so on. So that was a lot of the work, and actually training the model was not so much, but I think we were quite happy to see that this picture comes out to us in an unsupervised way. And as we sometimes say in my lab, like if we see it, we can predict it. So the realization that there's this strong imprinting of age is in these images was really important to us, because we can now, based on these rich morphological features, train what we call a biological clock. So this is not a new idea that came from us, because people realized more than 10 years, 12 or 14, some time ago, that based on a rich multivariate data, for example, as much more well-known in DNA methylation, you can train a predictor of biological age by actually trying to starting to observe the chronological age of different individuals, and at the population level trying to predict that. So based on molecular data in that case, or in our case in image data, predict the chronological age of different individuals. So a 20-year-old brain might look different from a 40-year-old, and different from a 60-year-old, and different from an 80-year-old. And at the population level, we learn to predict this global pattern. But then when we make precise or individual predictions for a specific donor, we may observe that the predicted age, so here in the y-axis for this, for a specific donor, may be different than their known chronological age. And the key here is that these errors, so these residuals in this regression task, are not, well, for the population, they are random, but they are meaningful. So individuals which are predicted to have an age that is above their chronological age, so this difference here we call an age gap, have been shown, for example, in DNA methylation data to fare worse in multiple health outcomes or in associations with disease and so on. And so we repurposed this idea to make our predictors of biological age from images, which was an entirely novel concept. So we call them tissue clocks. And we were successful in making these predictors of biological age for most of the tissues we looked at, essentially virtually all, just some we had a lower sample size, but conceptually there was nothing preventing us from doing it for all of them. And we had about an error of about 5 years for most clocks. So here we can see some insights that come from these models. So on average, we have about 5 years of error. And this still includes some parts that is biological, some parts that is technical, etc. But we are certainly doing better than sort of a random control. So this is just a sort of a technical control. The more interesting part is when we look at this deviation between the predicted age and the chronological age, so what we term an age gap. So individuals that have an increase in biological age compared with their peers of the same chronological age, so this positive age gap showed shorter telomeres, and this was measured in similar tissue blocks, but orthogonally, so in the same cohort, but with a different assay. But also interesting that if we look at these pathologies that are annotated in the image, so for example, if I'm 60 years old and I have a bit of liver fibrosis, doesn't mean I have liver disease, but this, what we call subclinical level of pathology, is something that is quite important for us. So these annotations of different levels of pathology that are in the different tissues, even though the individual did not have disease necessarily in that organ, was quite interesting. And we did see an enrichment for a more abundance of these pathologies with higher biological age gaps. So this is a very condensed representation because I'm looking at all tissues. And yes, there was some variability in this enrichment in different tissues. But also, if you look at the donor level, the number of comorbidities, for example, hypertension and diabetes and this type of common stuff that I already mentioned, this population is a sort of a representation of a general population. So some level of comorbidity was there. But even if one organ was affected, the others were collected, so the tissue we were looking at was still healthy. And we do see an enrichment in comorbidities in the number of and in the incidence of specific comorbidities with accelerated aging, so with higher age cap. So this was something that took us quite some time to believe or to have the right controls and to double check, etc. But I think something that really helped us a lot was to try to get to the interpretability of this. What is in here? What is conditioning, or what are the tissue-specific features that allow us to predict biological age in contrast with chronological age? So as an example, I have here decoupled the chronological age and the biological age acceleration. So on the y-axis, I have the gap, and the x-axis, I have chronological age of the different donors for one tissue in the aorta. And you can see that the ones that show most accelerated aging, as measured by our predictor, do look a little bit different, even if you don't know maybe what an aorta should look like. But this was interesting for us to evaluate. But we wanted to be a bit more systematic, right? Of course, we can look there and you can say, as a pathologist would, there's more of this, or there's less of that, or we could have set a trial with pathologists to look at this independently. But we have 25,000 whole slide images, so it's just not possible. And this is where the multimodal models were also very, very important for us, because, as I mentioned, they understand both the semantic context of the language of the images that they were trained together with, but the images they were trained together with were also very relevant because they had different pathologies. So in these models, you can query bidirectionally. You can have an image for which you want a text description. So if you have a text decoder, you can generate some text that describe it. Or if you have some text terms that you want to know, is it cancer, or is it benign, or is it a blood vessel, or is it a muscle? You can actually store these different text terms for images that you have. And this is actually how we did it. So we assembled a set of text terms describing different histologies and pathologies, and we basically saw here is a validation whether terms that we expected, for example, endothelium is higher in arteries or GI tract and so on. Adipose tissue is only adipose tissue, and breast muscle is predominantly in the muscle, skeletal muscle, but also in the heart, and so on and so on. So neurons is very specifically only in the brain. But of course, there's more terms that we can ask, or we can talk about sort of with a model. So, for example, if we ask what is the most, so some of the terms that were changing most with age, for example, in muscle, have to do with fat infiltration. So here you can see some specific examples of two images that are here. So this kind of more normal looking muscle. But here, the muscle fibers are much more further apart, and there's actually adipocytes in between, for example. In the nerve, there is atrophy. So you can see that the nerve bundles here are a little bit less tight and less packed than in the sort of in a healthy nerve. And loss of vasculature, for example, in the uterus. But there were a number of text terms that really helped us understand a little bit more systematically what are the changes that go along with age without, going manually through the images. So with this at hand, with some handle on interpretability and knowing that the age gaps are also relevant, we wanted to address a few questions. One of them was the question of organ specificity. Since we had multiple tissues per individual collected, so can we identify people whose heart is more accelerated, shows signs of accelerated aging, but the rest of the body is sort of average, or not particularly accelerated or decelerated? Or can we identify individuals who have a more systemic pattern of organ aging. So no matter what organ we look at, there's always biological age estimate, which is above their peers of the same chronological age. And can you identify some factors that condition this in a tissue-specific manner? Sorry. So on the first case, these are some examples. So each row here is one donor for which we have multiple organs or tissues. And we do indeed find several examples of people whose deviation is, for example, consistently below. So we call this the resilient agers, or people which are more or less on average. Most of their organs have an age gap around 0. It's people that have a single tissue that is more aged than the others, or a few tissues, and some indeed are systemic. However, most donors of the 1,000 we looked at actually did have indeed, let's say, a more in sync system. So the case where there was just one or few single tissues out of sync were more rare, but we did find nonetheless about a third of individuals, so 300, which had an age gap about 3 standard deviations higher than all the others. And within these, about half did have their body more or less in sync. So if one tissue was showing signs of accelerated aging, most others also tended to follow suit, and most of them were accelerated. So these are the pan-tissue agers, the systemic agers as well. It's the group in the middle. But then we did find also the hard agers and the adipose tissue kind of agers or combinations of organs that were, also immune system, spleen, skin, etc., that were a little bit more rare, but there are some combinations of people which we found like this. And we also, as I mentioned initially, we had some factors, both demographic, lifestyle, and clinical, for this cohort. Not all of them we could do a lot of statistics on, but some of them, about these ones, were recurrent enough or dense enough that we could actually test and see whether they accelerate or decelerate, protect from aging in a systemic way or in a way that is tissue specific. And by and large, they also tend to affect multiple organ systems. So this is maybe not too surprising, knowing that we also saw that most people were synchronized. But there were a few things that were a bit more specific to certain organs. They're highlighted, some of here. Some of the things that we found is that renal failure and sort of kidney disease was one of the strongest effect sizes in our cohort. But we also found, some things that are corroborative and nice to see. That if you have type 1 diabetes, your pancreas is about 6 years on average, kind of more aged than people of the same chronological age. But some that are also more surprising. So, for example, we did not know this at the time, but apparently people that have chronic kidney disease and have it's severe enough that they need, for example, dialysis or and have renal failure tend to have also peripheral neuropathies. And it's not quite known necessarily why, but we actually recovered that in the tibial nerve of females in this particular. So if it is separately per sex, actually have more accelerated age in the tibial nerve if they have renal failure, which is something that we're looking into, but also kind of interesting for a follow-up.
There was nothing like alcohol and liver age or anything like that I was looking for.
Yeah, so we do have some information on alcohol and smoking, but it's not very quantitative and necessarily reliable either, or we don't have a way to know it. So we decided not to include it here. It was pretty noisy.
I mean, one thing you could also think about is you could have a plot of like, what are these medical conditions and what's the average life expectancy from something like UK Biobank, maybe. It might be another way to correlate if your age gets too low, eventually you'll die from it, maybe. I mean, heart attack's definitely a good one. People die younger if you have a heart attack, yeah.
Yeah, that's true. Thanks. Just to wrap up this story, okay, so we were happy with all these insights, but if you it takes, a sample of your liver to tell you how old you are. It's not a very practical circumstance, right? So it's just not very applicable clinically. So something we thought was to leverage the breadth of data that this cohort has, and a lot of it, or some nice part of it, is in the blood. So we do have blood that was collected at the time of death or just before, and we have RNA expression that was collected from their PBMCs. So we thought, what if we would train something that from the blood of these donors learns to predict not the age of the donor or not also the age of the organ, but actually the deviation, so the age gap that is tissue specific. And we were quite surprised to see how initially things worked out, or seemed to, because once we predicted from blood the biological age of different particular organs, we actually saw that we still enrich for the pathology that is specific to that corresponding tissue more often than not. And also, for example, I guess really the breakthrough that came was that now that we are kind of free from tissue, so to say, that we have basically a blood based readout, we can go to external cohorts and validate this at more length. I don't have too much on it here, but just to kind of give a flavor on what we're seeing is that, for example, if we take individuals with Crohn's disease or rheumatoid arthritis, we already looked at now 8 different chronic conditions and some acute that recover accelerated aging derived from blood, specifically to in the organs that are known to be affected, but also in some others that, again, the kidney and the liver, maybe that might be due to compounded effects of medication and so on. But I think this is quite exciting. So the take-home message for this part is that aging has a profound effect on tissue structure of several organs, and that pathology really gives us a scale, together with deep learning and the depth and the sort of granularity and the richness of the features, to really study aging in humans. And that I think this can also be used practically for novel strategies for preventive medicine or for diagnostics, maybe drug discovery in the future, who knows. So we have some minutes to talk about multiplexed imaging. First, I'd like to say that, well, it's really an extension of immunofluorescence imaging. There's multiple ways of detection, but it really started with fluorescence, that we were able to capture multiple fluorophores or multiple epitopes, therefore, in tissue. There are constrained by the number of fluorophores we can capture by the wavelength, and therefore it gave rise to several people that were looking for strategies to get more and more multiplexing out of tissue images. And one of them is things like cyclic immunofluorescence or codecs as well. Here I'm just going to keep it kind of fairly agnostic to a specific type of assay, but they're all kind of conceptually similar. The idea is that you still only use, for example, 3 or 4 fluorophores, but there is different cycles in which you incubate with a set of antibodies, you stain, you image those, but then you bleach such that the fluorescence is terminated, and then you repeat again with a different set of antibodies while trying to not move the tissue as much as possible. So in this particular assay, it's kind of something anyone could have done in the lab. So this was developed quite some time ago. It's called CISIF. But there's many others like Codex and others that nowadays provide also like a commercial way to do it, and a device that can automate a lot of this. And the really nice thing, in particular of CISIF, is also the customization, because here, for example, you can see that there's for registered image, H&E image, There is a low power field here, and we're looking at a large tumor, but then there's still fields of view that if you want to acquire at 60x, you can. You can see here the outline of the nuclear envelope and things like that. So it can get really, really nice data with this type of assays. Here we're looking at images from codex. So on the left, we have for the same region of interest many different epitopes. So usually this can be nowadays expanded to 40, 50 epitopes. Maybe there's some assays that go up to 100, 200. Whether cost then becomes also an issue, but it's really empowering to be able to profile these many epitopes in a single piece of tissue. It has been for oncology, certainly, but also from other fields, as I will also talk a little bit about COVID-19, for example. But before going there, maybe I'll guide you a little bit through the computational part of analyzing these data. So particularly, key step is segmentation, which usually can be a semantic type of segmentation or object localization. For example, here we have in one case finding the boundaries of an object or look at kind of a bounding box. But if we have multiple instances of the same class, we can do the same task for each instance of that class. So if we have cells, you might want to find all the single cells or different instances of a cell, but also different classes of structures and so on. And for the longest time, this was not a very trivial task at all. In fact, when I started Cornell, this was still a big challenge. Nowadays, thanks to I would even pioneering tools like what I'm showing here have really accelerated and empowered biologists and pathologists and so on to do so much more with their data. So this was maybe the first generation of tools, and then when deep learning based tools like DeepCell and CellPose, etc., again became much more appealing to their ability to generalize. Yeah, so we are able to, from a single tissue section, collect 30, 40 different epitopes and profile the enormous diversity in the molecular variety of cell states, really, that are in tissue, but also in terms of structure. So we can see here, we're both on the left and the right side, we're looking at a lung, a human lung. There's still diversity along the lung in terms of the structure are there, but these images are maybe for our eyes not even almost recognizable as the same organ. So it really is important to have also enough molecular depth to know what we're looking at. So in this second example, I'll be talking about COVID-19, which we're probably often all familiar with. So in this case, it was a study that we ran very, very early in the pandemic. So we started in 2020. We were looking at the most severe spectrum of the disease. So people that unfortunately died from COVID-19, some that died after 1 to 5 days of disease onset, unfortunately, because it was the very early days of the pandemic, and disease was quite virulent, but also we were not quite empowered to deal with it very well. And people that, despite having severe disease very early on, were, for example, intubated for a long time in the hospital and eventually succumbed after a while. So we had this interesting axis of disease progression that we only had access to because of a rapid autopsy protocol that was in place at Cornell as well. And we had also looked at or assembled samples from other lung pathologies like sepsis, bacterial pneumonia, influenza, and so on. And we have this 37 marker imaging mass cytometry panel, for which we had such precious samples that we imaged as much as we could from these images. So we had 10 different regions of interest, about 1 millimeter square each. And we started exploring the data set. I think being a biologist by training and then also someone that cares about the systems level view of things, I was quite struck when I saw the plot on the left that I got the clinical data from our collaborators, and I saw the lungs had doubled in weight. It was not necessarily COVID specific, but I got very fascinated by this, and I started to conceive how to approach this problem from the top down, starting with the macroscopic observations and going more and more in detail. And the few things that we started seeing, just browsing through the images while we were trying to segment things properly and so on, was the amount of, the lack of space in the lung for, air and gas exchange and maintain the proper structure of the lung. So we could quantify that this lacunar space, these holes here that should be filled with air or blood, were severely reduced in COVID-19, in other pathologies as well. It was more severe with late disease. But in particular for COVID was what we saw, the position of collagen that you can see here on the right side was really dramatic, especially in late disease as well. So we started seeing like these macroscopic patterns of what was happening in the lungs, but also, of course, at a single cell level, once we phenotype, basically assign the identity to all the cells based on these markers, we also saw that there was a lot of immune infiltration, as I'm showing here. Initially, it was thought that the major immune cell infiltrating the lung were neutrophils because it's what I had seen in the blood, but it turned out to be macrophages, and in particular interstitial macrophages. So it's really important. This was highlighting the importance of looking at the tissue in very severe disease when possible. And another really insightful, in retrospect, decision we had when we were designing the panel was to include an antibody to the epitope itself, to the spike protein of SARS-CoV-2, which had just been made very, very recently. So this allowed us to, within one single image, to pinpoint not just the cell types that were infected, but also, for example, to observe which markers were upregulated once the cells were infected, or to see some patterns of avoidance or tropism in the infected cells. So this was quite novel at the time and really not done very, very much at the time. Of course, what we can do with the spatial data very well is to also observe the patterns of cellular interactions. So for example, here I talked about viral tropism. So this is basically the heterogeneity in the spatial manifestation of the infected cells and so on. We have infected cells that are interacting with non-infected cells, with immune cells, with itself. We have areas of concentration in infected cells. We have areas that are more sparse. I'm missing a plot here on the right, sorry. No, yes, it's coming. And by quantifying these differential interactions based on infected cells and not, we could also see interactions that were driving the more later stage of disease, for example, between macrophages and fibroblasts that were really driving the fibrotic phenotype. Even after the virus gets cleared, there's the pathology continues manifesting because the immune system is overactivated, in particular these macrophages and so on. And lastly, something.
The next speakers here, we might have to go a little bit, finish up in the next minute or two, I think.
Right. So I'm really at the end. Thanks. Okay, I will continue. Yeah. Something that's quite important to me is really to also put these observations in a clinical and a biological context and not just stay with, sort of an atlas view that for the sake of studying cells. So this is something that we also try to do by incorporating both the clinical data and that came from pathology, as already mentioned, and characterization of the lung tissue on the clinical course. And we helped us also find different clusters of donors that based on these different phenotypes and their pattern of progression. So this is the second part. Regarding the frontiers, if you give me one more minute, I will really just say a few words. So the first one is really an obvious one, is the molecular depth in which we need to profile tissue spatially. I'm showing here an example of StereoSeq, which in this case is also a nice example of time resolution and so on. But I think that something that we need to work towards is also an understanding cross-scale biology, going from single cells to higher order structures and whole organ, whole organism. And I think the project that I showed on the histology is also an interesting way of how can we combine insights from different organs. Hopefully going in even beyond that, because combining also things like CT and MRI, I think it's also quite interesting. 3D is also an avenue that is in active development. It's quite demanding, but I think it will be quite interesting. And really the one that I think will really open up more avenues in the future is also going to be in vivo or less invasive type of imaging or type of readouts for proxies that are tissue specific, phenotypes that are tissue specific, as also I've shown you in the work combining blood data and tissue phenotypes. And lastly, I think if we are successful at all of this, we can also be better at doing kind of generative model and regenerative, perhaps, medicine based on designing new tissues with certain properties. So thank you so much. If there is some pressing questions, I'm also happy to address. And sorry if I got a little bit over time.
Thank you. Any questions? Time for maybe a quick question. I'll throw one in. I think you noted it's hard to take a biopsy from someone to tell their age, but you looked at other metrics to infer it. Have you looked at cell-free RNA or cell-free DNA where you can infer like recently lysed tissues or other ideas like that? And curious if that would help.
Yes, it's something we're thinking of, particularly in light of recent papers and also in the work that is going on in plasma proteomics.
Yeah.
I think that's something that we're capturing in the blood data. So I think we got a bit lucky because in this cohort, the data are sequenced very deeply. And I think we have captured also some sort of ambient RNA because we can, of course, know which genes are giving us the predictive power, and they are for sure not expressed by immune cells. So we are doing work on that too.